Hyperloop? What's so special about it? And the answer is crystal clear. Because Hyperloop has too many advantages. Like, Hyperloop can save an enormous amount of traveling time. It is eco-friendly and it operates at reduced flight cost and time. Emissions like GHG will be talk about its innovation and contribution and its structure. So there people can travel capsules. So there are many capsules, 6 to 8 capsules per 3 capsules per train. So its unique character is it runs in induction motor instead of wheels, and it runs in a vacuum channel. Smart AI control systems for autonomous capsule management. The capsules are pulled along by magnetic levitations. On-road trials have been there in many cases like the Virgin Hyperloop 1, Hyperloop TT, Zero Loss and many more. Each capsule floats on a cushion of air as it creates and moves forward. To minimize the friction, a powerful fan at the front would suck what air is in the tube to the rear. There are many Hyperloop routes in India like the Delhi-Amritsar routes, Mumbai-Pune routes and many more. The use of Hyperloop in commercial fields is expected to be seen within the next 10 to 20 years. Hyperloop is the 5th form of transportation. Its minimum speed is 1200 km per hour. So if we see that what is the function of Hyperloop, so it uses the induction motor because to minimize air resistance, and Hyperloop is likely generate more power than it consumes. So if we use the mode of Hyperloop to travel, it will make us travel in more time. And if we compare to many things like the bullet train or Boeing, so the Hyperloop has higher speed but with too many advantages. So we come up about its disadvantages. The disadvantages is very less in comparison to the advantages. So if we compare the disadvantage is that it occupies land acquisition and the construction cost is low, so that the Hyperloop has not been a trend in nowadays. Thus, Hyperloop is a combination of speed, sustainability and innovation. Thank you.
Anybody want to ask question? Okay, my question is there. What is Hyperloop? What is the main principle behind your Hyperloop?
So the main principle is to minimize the time and to enhance the efficiency. The Hyperloop reduces the traveling time and conserves the energy of the mechanism.
If that is the mechanism, we have to travel with a minimum speed of 200 km per hour. And just to minimize time, they have designed this. And if accident cause, you will never reach your destination. I just asked what is the principle behind that. We are not spoken about the pressure pipes. We are not spoken about the magnetic cords. We are not spoken about the pressure releases at the front, lower and back, which makes it floating on the way.
So the pods are regulated magnetically on the air bearings. And
it's like floating like a thing, or it should be a curved arch.
These are the pointed structure.
Tunnel will be passed by. Okay, thank you.
We have a gentle reminder to all the participants. You all have a maximum of 5 minutes for your presentation. So kindly ensure that you stay with the last set of questions. Next we have Arisana Mahanti from Stewart Science Higher Secondary School. Please welcome her with a warm round of applause.
Let me make you understand what is the scoring system. In scoring system, it is total of 20 marks, out of which 5 5 marks are for visual, 5 marks are for content, 5 marks is the mode of delivery, how you deliver, and 5 marks is audience engagement on your defense. Okay, in that context, we will be 20 marks.
Good morning, everyone. Now imagine one of your closest family members had an organ failure and they were waiting at the hospital searching for an organ transplant. Now, annually over 100,000 people face this in the US alone, and out of which 70 people die on a daily basis. Now to tackle this scenario, scientists and researchers at Harvard University and the New York University conducted a research where they used xenotransplantation with the help by transplanting pig's organ into a human body. Now, the issue here was that the pig's organs had harmful pathogens such as the alpha gas, which causes hyperactive rejection in the human organ. Now, to tackle that, they use CRISPR-Cas9 gene editing, which is a process which acts as a harmful DNA from the pig's organ to act as a more suitable organ transplant for the human. Now, the issue here was that even after cutting that off, the organ still showed a failure after 12 hours or 48 hours of the xenotransplantation. This was due to antibody-mediated rejection, which was shown by the human immune cells and the antibodies present in the human body. Now, to tackle this, single-cell RNA sequencing was used. A biopsy was taken which contained both to take out the particular cell characteristics of each cells. Now, the single-cell RNA sequencing did do that, but it failed to dissociate between the human cells and the pig cells. Now, to tackle that with the help of bioinformatics, researchers made a computer file which was the hybrid reference genome, which contained that particular cell into either of these categories, either the pig cell or the human cell. This was done because the human cells present in that biopsy was less than 5% of that compared to the pig cells. Now this was done because at the 12-hour mark, there was a steep rise in the human antibody generation, and at the 48 hours mark, it was also shown that. Now this gave the researchers the answer that at the 12 hours mark and the 48-hour mark, the human cells showed immunity system and which attacked the pig cells. Now another fact was shown that the IF and gamma chemokine was which acts as a signal which shows that the human cells have started to form, have started to attack the pig cell. Now this paper suggests the genetic engineers that to solve this particular issue, they have to insert a CD47 gene into the pig's organ to help solve the antibody immunization and to help solve the hyperactive rejection. Now, this helps in a way in the future when there is a lack of organ transplants, they can use a pig's DNA, a pig's organ transplant into the human's body. Thank you.
Any questions? Okay, what's the name? In a layman's voice, what do you think?
So the layman's voice has said that so basically there is a organ shortage crisis globally because a lot of people have organ failure and they are waitlisted at the hospital or they die due to the lack of organ transplant. So to fix that issue,
So what your topic says that a pig's kidney can replace a human's
Yes, sir. So that is a
Now, I'll ask one question. Can you say that your kidney can be replaced by your son's kidney?
so technically yes, sir, but
No. It is not technically yes. It is absolutely no. For any transplantation you need inside body, no. First there is a rejection problem. If at all you put steroids, I think it is a day recently before 5 days something a mother and a son both died after transplantation of the kidney. The problem was the mother was having B positive blood group, the son was having O positive blood group. Still, they are same. They are 50% of the reasons she donated, but both of them died. It's just 50 or 5 days before. That is on Facebook. See, there is an organ rejection included when you do not match your blood group. If at all you put steroid to suppress or minimize your rejection, still when the effect of steroid is over, the rejection will continue. So now the second question is from where you got this idea of
has been this research has been conducted at Harvard Medical School with the professors from Harvard Medical School as well as MIT and as well as Yale University. And all it does is, and I actually used an existing research article as a
now we just read the lower one of cellular dynamics into human kidney transplantation, focusing on single cell genomics and hybrid genome tracking. They are not changing the organ data, they are just talking about the cell differentiation which will occur between the human and the pig. They are just trying to find out. See, I will say you one thing. There is one Dr. Jayan Das in Bhubaneswar. He is doctor at some ultimate go at meeting, ask me name. He had done a bank of skin as well as organ. He is doing it personally. You check on Google, Dr. Jayan Das. Anybody can check. The motive here is they are trying to imitate human cell from a mammal cell. Now, in case of mammal, pigs are those mammals which grows at intense speeds. That is why they are taking pigs. And they are trying to find out a genomic concept, like you have done human genome project, but they are finding out the common part of human and a pig so that they can develop a skin, a cell, or an organ by chance in near future. Okay? So, our topic that also पूछो तो जो अन्य वाला टॉपिक नौ पूछी गई पहले भी कॉल पर सिला बोले
so the thing is that I didn't understand the topic, but I was not able to explain it correctly. But I have understood.
Next up we have Devoshita Nanda from Mahamati Vihar Women's College. I request her to kindly come to the podium and present her. Please welcome her to the class. Good morning, everyone. And my seniors. So today I'm going to present biological insights and computational intelligence. So first we will know that what is the bioinformatics for the field, and then biology means technology to discover the course of life. We are living in an era where science has the potential to improve. Science has the potential to improve our lives in an unimaginable way, where the bioinformatics is the key for that. So first of all, we will know that what is bioinformatics. Bioinformatics is the interdisciplinary field that uses computer science, statistics, and mathematics to analyze or interpret large complex biological data sets like DNA, RNA, and protein sequences, and it involves developing software tools and analyze or algorithms to store, retrieve, and and store, retrieve and analyze biological information such as with the playing a crucial role in the cell, like genetics, drug discovery and personalized medicine. After that, we will know that what is the history and scope of bioinformatics. The history began with the networking of protein sequences or early protein sequences from the analysis by the first biological analysis like GenBank in 1970s. Force since expanded dramatically, driven by the Human Genome Project. Advances to sequencing to include the data from molecular analysis to organismal level, with applications ranging from personalized medicines, drug discovery, and to evolutionary studies and analytic or synthetic biology. And the scopes are that the terms bioinformatics was officially coined by Pauline Hobbe and Ben Hispa in 1950s. The first protein sequence, that is bovine insulin, was published promptly as the computational analysis. After that, in 1974, DNA GenBank that was initiated by a computer and software like GCG, which allows more complex analysis of bioinformatics. After that, in 2003, the Human Genome Project was completed, and huge data produced by this needed the bioinformatic tools to interpret and analyze the complex databases. So, after the what are the methods of HGP? We know that methods of Human Genome Project contains two aspects, that is sequence annotation and express sequence tags. Sequence annotation, where both coding and non-coding sequences, are identified, and later on they are assigned through their respective functions. And after that, express sequence tags, where the DNA are identified or expressed themselves in the form of RNA, are identified here. The piece of DNA which are identified is isolated and associated with the vector DNA, and then introduced into the host, which is amplified inside it. After the amplification of genes, it is easy to sequence them. The vector DNA may be an artificial chromosome or bacterial artificial chromosome. So after that, we are going to know that what are the silent features of Human Genome Project. The Human Genome contains 316.7 million nucleotide bases. To average nucleotide bases contains of 3,000 base pairs, but the maximum up to 2.4 million bases, which are exactly the same. The average base pairs or nucleotide base pairs are estimated as 30,000. After that, we are going to know that what are the applications of Human Genome Project. The exploration of human genome opens a new approach in biological databases or biological research. The knowledge about the effect of the DNA, can revolutionary and new ways to diagnose, and it helps in the database of cells like precision in medicines and agriculture and drug discovery.
Tell me the relationship between the Human Genome Project and bioinformatics.
So, we have Indra Patnaik from Mahanadi Vihar Women's College. Please welcome her with a warm round of applause. Myself Indira Patnaik from Mahanadi Women's High Secondary School. Today I am present here to deliver a talk on bioinformatics, the convergence of biological insights and computational intelligence. So from the word we can know that the bioinformatics is limited towards bioinformatics. Here bio refers to biological systems, organisms and data related to it. And informatics refers to the computational tools and methods used to analyze and interpret the data. So we can say that bioinformatics is defined as an interdisciplinary field that uses computational methods and tools to analyze and interpret biological complex data sets such as DNA, RNA, protein sequences. So now talking about the areas of the bioinformatics, it has a wide range areas to focus on like genomics, proteomics, cheminformatics, computational biology, and also systematic biology. We also have some key experts. The experts are crucial to understand the complex biological relationships between the organisms, molecular structures, protein sequences. Some experts are interdisciplinary nature, and also data management, storage and retrieval of the data. Also application area and processes are involved. Now, moving on to an important point in the slide is that how the HGP and bioinformatics are interlinked. So, as the 13-year effort of mega project, HGP, or you can say the Human Genome Project, led to the coordination of many countries like USA, UK, China, and also many scientists from all over the world. The project generated a huge amount of data, which manually would have required at least 3,300 books with 1,000 minimum pages, which was quite risky because maybe due to some natural disasters the books have missed. So that was the point of this, to store, save and manage the generated data, the biological data by HGP properly. By this we can know that how HGP and bioinformatics are very closely associated, or you can say the internet. The connection can be understood by several ways like data management, as I discussed earlier, that computational tools are provided by the bioinformatics. Also, we have development of many tools as the need to handle the HGP data required and led to significant tools development. And we have advancements in biological and healthcare sectors. And lastly, moving on to the applications of bioinformatics, it has applications in many areas such as agricultural and environmental fields for crop and livestock improvements, also some biotechnological products. And we also have discovered drugs and personalized medicines, also gene therapy is included. So that is all the presentation. Thank you so much.
No questions, I guess. Please.
So my question is that how does gene therapy help us?
Can you repeat the question, please?
You said that gene therapy is helpful to us as an application of bioinformatics. So my question is that how is gene therapy useful?
The answer is bioinformatics provides bioinformatics?
Bioinformatics provides computational tools to analyze and interpret the data, and computational biology uses those tools to analyze and solve the problems.
Can you give some example of bioinformatic tools?
Like bioconductor and software like BLAST.
Bioinformatics is not good. Last is
Yes,
particular question, bioinformatics never take any, what you say, any kind of wet lab decision. They are not got anything using bioinformatics. But we predict. There is a prediction where there is a low chance of any human loss. That is what bioinformatics. Okay. Okay. Tell me one thing. Storage and retrieval application areas, core processes. Can you please explain those three parts? Storage and retrieval applications area and core processes. What is the core process over here?
the core processes involve like identifying the genes and also predicting the protein structures.
Data analysis.
Yes, sir.
Then what is core process? You have already mentioned data analysis. Data analysis means right.
Processes like identifying the genes and protein structure predictions.
Okay, thank you.
That was indeed a wonderful presentation. Now I request Maitreya Abhilash Patnaik from Stewart School, Bhubaneswar to kindly come to the podium and present his presentation. Please welcome him with a warm round of applause.
Carl Sagan once said that the more we learn about ourselves, the more we know how we are related and connected to the species who are alive, who are present on Earth. Good morning, everyone. I am Maitreya Abhilash Patnaik from Stewart School, Bhubaneswar. Today, I am standing on this esteemed platform to speak on the topic comparative genetic analysis of humans and primates. So, this is a topic which links biology, technology, and our identity as humans. So, when scientists compare the genomes of primates and humans, they found a very amazing thing. They learned that humans share 98 to 99% of DNA with chimpanzees, 99% with gorillas, and even with species of monkeys, such as the rhesus monkeys, which share like 99% of their gene and DNA with us. What does this prove then? This proves that all of us, the primates, had a common ancestor millions of years ago. Therefore, we have such similarities. But then a question might arise in your mind, which is that if we are so alike, we almost share like 99% of our genes together, then why do we look different? Why are we more advanced than the other primates? Well, let us look at some of the key differences one by one to know why we are so advanced. First is chromosome. And other primates like chimpanzees, gorillas and apes, they have 24 pairs of chromosomes. But in humans, they have 23 pairs of chromosomes. This is because during ancient time, when our ancestors split up to give rise to the human species and other primates, the two chromosomes which were ancestral chromosomes which were chromosome 2A and chromosome 2B, they fused to form the present chimpanzees, it is found that human brains are 3 times as large as compared to chimpanzees, although we have the same DNA. It is because of small genetic differences which can lead to such big changes. Then movement. Apes, chimpanzees and other mammals and other primates show quadrupedalism. That means they usually walk on 4 limbs. But in case of humans, we show bipedalism. That is, we stand upright, we walk upright. We use only 2 of our limbs walking upright. And why does this happen? Because of changes in our skeletal system and due to gene regulation. Next. Now, why are we here? Now, what does the science society benefit from this? What is the purpose of this? What is the purpose of this genetic analysis? By this genetic analysis, scientists came to know that even a small change in DNA, one small change in gene can make very big differences in humans. Such as a very small change in gene has made humans and chimpanzees completely separated. Next, by studying the genome of apes and other primates and comparing them with humans, we can understand the reasons for which gene is the cause of the certain diseases which humans can acquire, but the other primates are not infected by it. Next, so it will also be helpful to find medicines for many diseases. Next, I want to conclude on the note that this comparative genetic analysis shows that humans and primates are not 2 different creations. They have arrived from the same evolutionary tree, and our DNA reminds us that while we have reached so high, we have become so advanced, but we remain deeply rooted in the living world that shaped us. Thank you.
Any questions? Please come here. Because I have to come on the
So you mentioned in the presentation about comparing humans' DNA with chimpanzees and other primates. So according to basically Neanderthals are more closely related to us than chimpanzees and other primates. So why is it not that Neanderthals were taken into consideration into here? Because they are more closer to us than other primates and they are relatively more easy to compare to us than other primates.
First of all, it is because that Neanderthals are considered as a subspecies of the humans. They were to learn about them, and it was easier to select other primates like chimpanzees and gorillas because they are not extinct, they are currently living. So it will be easier for us to study them and compare them with humans as possible.
Please try to remember this. There is only movie Fish where Hindi Ocean plays the father's school and something. There is no truth about that. And the question what we ask from Neanderthal human, you are not saying you have 2 types of genes, guys. Please note this. One is called complementary genes and other is called supplementary genes, which works on according to the environmental system. I'll let you give a golden example. In India also, you move throughout the country now, you will mark the difference between groups. If you go to southern part, if you go to western part, if you go to northern side like Punjab, where they have good physical tin, which is a muscle protein, helps in muscle development. Due to presence and secretion of this muscle protein, the apes are tetrapodal. They use 4 hands to work. Yes. And since we have only 8% means in human body, we are bipedal. We walk on 2 feet and straight.
Yes, sir.
Okay. Understood. Now my question is, tell me one thing. When you choose this topic, right, you have not mentioned that chromosome 2 is a muscle group. Why?
So I had researched on the topic, but it had not come under my consideration.
Okay, what innovation came to your mind to
this one more interesting because we were comparing ourselves with another species, so it was more realistic and more easy to understand.
Yes, that is true. And this is why research still ongoing after human genome.
Yes.
They have not stopped yet. They have included 22 countries in whole world except India. Except India, they included 22 countries in the whole world. And this is a part that which human being developed two vaccines for COVID. One is called as Covaxin, other is called as Covishield. Both the vaccines were developed from one was developed from the from the immune system of the chimpanzee, other was from the his surface protein of the chimpanzee.
Yes.
That is why they have two different types of solutions. Thank you.
That was indeed a wonderful presentation. Now I request Manushmita Senapati from Stewart Science School, Bhubaneswar, to kindly come to the podium and present her presentation. Please welcome her with a warm round of applause.
When biology meets bites, discoveries happen at the speed of lightning. Good afternoon everyone, I am Manushmita Senapati from Stewart School, Bhubaneswar, and today I am going to speak about bioinformatics and especially its application, which is drug test. It is really exciting and fascinating when biology and technology fuse together, especially in the field of medicine and science. So biology and technology fuse together to help us in various safer and more effective. So today our objective is to learn what bioinformatics is, its applications, especially drug tests, and few of its examples. So bioinformatics is basically like a very big book which contains all the instructions about our life. It's such a vast and complex book that we need AI and technology to decode it. Bioinformatics combines maths, computer and biology, and it helps us to analyze genetic data. Yes, sir. So I chose drug test because drug test is a very vital application in the field of bioinformatics because it helps us to know how our medicines are made, how the vaccines are made, how it's effectively made and provided to us. It has very long processes. The first process would be the virtual screening. In virtual screening, AI identifies potential participants to be tested on. Here, many pharmaceutical companies use this feature to find potential treatment for many diseases like neurological syndromes and specific cancers. Then, preclinical trials. In preclinical trials, these drugs are tested on cells of various animals. An example for this would be AI helped us to optimize a very good drug for breast cancer, which is palbociclib. Then clinical trials, which is the final process. Here, the drugs are finally tested on humans. And these drugs are then tested on humans for the final stage of the bioinformatics. Then, example for this drug test would be COVID-19. Bioinformatics has helped us very nicely to provide vaccine for COVID-19. Here, scientists use the AI features to study the virus, the protein structures, etc. So basically, bioinformatics helps us very nicely in the field of drug testing. When biology and technology mix together, it forms a dynamic duo which helps us to find various solutions for various problems. So basically, it helps us to provide medicine, which is a very big step in the path of healthcare. Thank you.
Can you please tell me that how AI identifies a cell it is cancerous or not? How does it identify?
The AI first sees the cells. On the basis of the disease, when it separates the diseases and it sees if the cancer cell is dividing or not, then it escalates it as a cancer cell or not.
कैंसर सेल ଯେତେବେଳେ ଗୋଟେ ସେଲ୍ କାଉଣ୍ଟିଂ ହେଉଛି ଆମର କଲୋନି କାଉଣ୍ଟର ମେକାନିଜମ where you can count your cell ଯେତେବେଳେ ବାୟୋପ୍ସି କରାଯାଉଛି what doctor or a cancer specialist do it takes some ତ ଗୋଟେ ସ୍କ୍ରାପ ରୁ ଗୋଟେ ସିଙ୍ଗଲ ସେଲ୍ କାଉଣ୍ଟ ମନେ କରୁଛ 50 ଆଜି ମନେ ଦେଲା 50 ଟା ଅଛି ଏକ୍ସାକ୍ଟଲି 32 ଘଣ୍ଟା પછી ମଣିବ ତ 50 କିନ୍ତୁ ହବା କଥା ମାନେ ସ୍କିନ୍ ରୁ ହେଉଛି ମାଇକ୍ରୋସ୍କୋପ ସବୁ ତ 100 ହବା କଥା ଯଦି 100 ରୁ ଅଧିକ ହେଇକି 110.105 ହେଲେ ଚଳେ କିନ୍ତୁ ଯଦି 150 ରୁ ହେଇଗଲା ତାହେଲେ କ୍ୟାନ୍ସର ଗୋଟେ ବାୟୋଲୋଜିକାଲ କରିବା ଏଇଟା ଭେଣ୍ଟାର ଗୋଟେ ବାୟୋଲୋଜିକାଲ ଟେକ୍ନୋଲୋଜି ଇଜ ପାର୍ଟ ଏଇଟା ହେଲା ପ୍ରେଡିକ୍ସନ କର କାଇଁ
Yes, sir.
Now, my question is, differentiate between a vaccine and drug.
Sir, vaccine and drug. Sir, drug is like a medicine which helps us to cure a disease or maybe suppress it after we have already suffered from a disease. But vaccine, we take it before we suffer from a disease to prevent it. Here, the
COVID do not kill us and vaccine alone.
sir, to prevent it further, the vaccine actually works on our cells, so that our cells actually identify the disease beforehand. Usually this happens so that our cells diseases and they fight for our immune system.
Second, you have given pre-clinical trial and clinical trial. Definition of those.
So in pre-clinical trials, these drugs are tested on animal cells.
Animal में born होगा, लेकिन animal active नहीं होते हैं। आपको animal में चूंकि ले होते हैं।
So other cells except for humans, because
I'll give you an answer. Vaccines are attenuated microorganisms. They are living microorganisms which are injected to the body
Yes.
before the age of 5 years. 5 ବର୍ଷ ବା 7 ବର୍ଷ ଭିତରେ 0 to 7 years 5 years ଭିତରେ we are injected with all viruses available on the data sheet. In HIV, hepatitis, flu, whatever, polio, whatever. So by the time you grow up to 35 years of age, that is the age when they will be infecting you, you will be having n number of antibodies in your body. So that when the original target comes out, you will fight it. Or original virus will fight it. That is the vaccine. Maybe a salt, maybe a steroid, maybe something. Okay? Thank you.
thank you, sir.
Thank you for your presentation. Next up, we have Karthik Patnaik. I request you to kindly come to the podium to present